Thank you very much. Being an employee of the EPA, I have to start with a disclaimer that the views I'm expressing are my own and not reflecting the views or policies of the EPA. I'd like to thank the NES Standing Committee for providing me the opportunity to provide some input on sort of the overall context from our perspective in terms of the perspective of this workshop series on taking emerging science to inform environmental health decisions. At the National Center for Environmental Assessment, we develop what are called health assessments or toxicological reviews of various chemicals that are in the environment. And so we're a potential user, essentially, of these types of data. I'm going to actually start by acknowledging the many colleagues who've contributed to the ideas and approaches that I'll be presenting, including scientists from EPA, other government agencies, academia in the US and abroad. So an outline of my presentation. Basically, I'll begin with sort of a general context in terms of human health risk assessment and environmental decision making, and start with some kind of overarching questions as to, for example, where do tissue chips does this work as a touchscreen? Oh, there's okay, well, anyway, where do tissue chips fit in the source to outcome continuum, how they might be used in hazard identification, dose response assessment, standard parts of our toxicological review processes, and then whether there are some key environmental health challenges that tissue helps can help address. And I've listed 4 of them here that I'll go into a little bit more detail on. So what is human health risk assessment? It really is how science is supposed to inform decision making. And on the right here, I've shown our recently published EPA framework for human health risk assessment, which really is a process in which science is embedded as the data that's being used, but it's within a broader context. So we take the available data, whether it's experimental data, observational data, various concepts, models, and those really need to be translated into a form that can be used to inform decisions. That requires evaluating data and analyzing, making extrapolations, which are often necessary, and synthesizing all the different lines of evidence to ultimately inform decision-making in a regulatory context and also in non-regulatory context. And this includes data that's been talked about for the last almost a decade now in terms of toxicity testing in the 21st century, new sources of data, QSAR, mode of action, adverse outcome pathways, in vitro screening, et cetera. So how do we translate this data? And that's really putting this data into a context that can be understood from the environmental health side. And then also at the same time understanding what the type of environmental health decisions we need to be informing. I've shown here just a number of the different types of decision making that can occur at EPA and at state and local health agencies, whether it's screening and prioritization, setting particular levels like maximum contaminant levels in drinking water or ambient air criteria, population level analyses where we're doing sort of looking what is the overall impact across a population that's exposed, as well as risk comparisons, comparing alternatives. And really what decision you're trying to inform needs to really drive the design of the risk assessment and how different information along the source to outcome continuum gets synthesized together in terms of how we design, how we measure exposure, how we identify hazards, how we do dose response, and ultimately how we characterize a risk from environmental contaminant or exposure to inform a particular decision. I'm going to focus on hazard identification and dose response since that's the most relevant area for tissue chip type data. So the first real question is, where do tissue chips fit in this sort of source to outcome continuum? So just as an overview for those who aren't as familiar of the environmental health context, really we're talking about going from concentrations in the environment or sources of emissions, looking at the exposure pathways as to how that gets to doses to individuals, going to, looking at the toxicokinetics to see how those translate into internal concentrations, looking at toxodynamics in terms of how internal concentrations translate into biological responses, and then how the system responds to those biological perturbations to ultimately affect one's physiological health status. And so the types of data we use in our typical assessments include epidemiology, animal bioassays, PBPK models, in vitro assays, looking at adverse outcome pathways more recently, and really covering various sort of parts of this continuum. So the natural question for tissue chips then is sort of where might any particular platform fit in terms of this source to outcome continuum. So that's sort of the first question we would want to understand in terms of how we integrate that and synthesize it with all the other data we might be considering. The second then has to do with in terms of looking at it in the context of the environmental health decisions. So a prototypical hazard identification and dose response will be looking at identifying and evaluating all the available evidence that's out there, identifying specific health effects that we believe there's a sufficient evidence for them to be considered causally related to exposure to a particular compound, for instance, maybe developmental effects or carcinogenicity. And then estimating what are called points of departure from suitable studies, that is low observed adverse effect levels or no ALs, or better yet, benchmark response levels, benchmark dose levels for particular levels of response. And then calculating what are called toxicity values, either something like a reference dose where you take the point of departure and divide it by factors to account for various uncertainties, or doing something more akin to a straight line kind of linear extrapolation to understand what the responses might be at exposures lower than can be measured in experimental or observational data. And then those toxicity values then are used in helping inform these environmental health decisions. So then where do tissue chips fit in each of these steps? So now the next thing I'm going to spend most of my time on is talking about some of these key challenges in environmental health. And that includes several that have been discussed in the previous talks, including the number of chemicals without assessments, trying to get to a higher throughput, addressing uncertainty and variability, which a number of NRC reports have over the last many decades have talked about, and that science and decisions referred to as sort of recurring and recalcitrant challenges in environmental health. And then the fourth one, which I think has not had as much attention in environmental health sort of scientific community, is supporting cost-benefit analysis from the economic side. And even though that really is a very strong regulatory driver, particularly at EPA for many major regulatory decisions. So for each of these, I'm going to identify sort of a number of key questions and issues as to potentially incorporating data from tissue chips. So the first challenge, in terms of there being many more chemicals and human health assessments, this is a challenge by any measure really. The gap is in the thousands, even for high production volume chemicals that have more than like 1 million pounds of production a year, and into the tens of thousands if you're looking at the whole TSCA inventory. And this has been a topic of discussion for many years. And this is a sort of augmentation of what Dr. Rusin presented earlier. A lot of the limitations in the available is both in the available data, but also in the methods for translating those data into a form that can inform decisions. So in the program that I'm in, National Center for Environmental Assessment, the Human Health Risk Assessment Program, there are sort of 3 general product lines that we have: the integrated science assessments, which look at criteria pollutants like PM and NOx and SOx and lead, that are really sort of recurring assessments of the state of the science of what the health effects from exposures to those compounds are. And those are primarily really based on human studies. There's the IRIS assessments, the program that that my branch is in, where we both look at human data if it's available, but more commonly chronic animal studies is the basis of our assessments. And then provisional peer-reviewed toxicity values that are used primarily by Superfund to look at Superfund cleanups. And that in those cases, there's generally less data, occasionally chronic studies, sometimes only subchronic studies. So then the question is, how can we get to higher throughput approaches, ones that can address many more chemicals using either in vitro or alternative species, using just inherent chemical properties, but then also developing the methods and approaches to sort of translate that data into an assessment that can be used in decision making. And this is sort of the area I think where tissue chips will probably be residing in terms of how they'll be supporting this challenge of not enough assessments and too many chemicals. So some of the questions that we would have in terms of trying to incorporate these is sort of what are some of the key existing limitations that you might be able to fill that gap with tissue chips. We need to understand the cost and throughput in comparison with in vivo data and even with alternative in vitro systems. What is their coverage in terms of chemicals? Can they address sort of ranges of solubility in different media, volatility, metals, some things that some of the other sort of approaches to higher throughput assessments have some limitations in? What's their coverage in terms of metabolism, being able to incorporate metabolism, which has also been a challenge in some in vitro systems. Look in the coverage of sort of toxicity pathways and the types of endpoints that can be covered. And then this was brought up in the previous talk about the data quality across and moving sort of into a commercial. Will these become commercialized in the sense that we'll sort of have a sort of off-the-shelf products so that we'll be assured of the data quality as long with specific protocols, et cetera, that can provide kind of a gold standard kind of QA, QC kind of thing. And then really a big challenge in terms of interpreting these results in terms of decision making. How do we maybe anchor to in vivo approaches or do some sort of parallelogram where you might have both human and animal on a tissue chip and then animal in vivo and then be able to sort of predict the missing corner there. A second big challenge is addressing uncertainty, and here I really mean addressing it quantitatively. And just as an illustration, so the typical way in which we calculate a reference dose is you start with a point of departure, which is based on the available data. You divide by a number of factors to get to a reference dose. Now, one of the issues here is that these factors, although they're called uncertainty factors in the US at the EPA, they're not really just about uncertainty. They're really a mixture of adjustments, of uncertainty and accounting for variability. And also they're presumed to be conservative individually, although in some cases the empirical data for how conservative they really are might not be terribly strong. On the other hand, if you have multiple factors together and they each are conservative and you're just multiplying them, that can lead to this issue of sort of compounding conservatism that has been raised by many people. So really, the RFD is supposed to be some level at which there's no appreciable risk, but really, we don't really understand whether they're all the same level of confidence as to likely to be without appreciable risk. How likely, are we at a 95%, 99%, 99.999%, et cetera, without some sort of more quantitative measure of uncertainty. And then additionally, some decision contexts really would benefit from more than just a conservative bound. If you're looking to choose amongst alternatives, if you're allocating scarce resources amongst different options, and also for economic analyses, which I'll get to later. For instance, if you were to have an entire sort of distribution rather than just sort of a conservative bound, you would, and then you have different decision rules as to whether you're going to decide on a lower bound, decide on a central estimate, decide on the width of the range. You could come up with very different priorities. So that sort of illustrates the need to understand the full uncertainty range, as well as to understand in your decision context, what is the appropriate type of decision rules that you would want to apply. So, this issue has been, raised for, at least 2 decades in sort of environmental health risk assessment. And one of the approaches that has been advocated is development of a more probabilistic framework for toxicity values. And more recently, in the last 5 or 6 years, I've been a part of a WHO IPCS project in which we've been developing a framework, this type of framework for moving away from an RFD type approach to a more probabilistic one. And a guidance document is actually in press this summer and should be available by the end of the summer. So the basic idea here is to replace something that's a little bit amorphous, like the reference dose, with something that is more sort of explicitly specified in terms of a target human dose. We call it HDMI, which people will more easily remember because it's that cable you plug into your HD TV. So the HDMI is the human dose at which a particular fraction of the population or incidence I shows an effect of a particular magnitude or severity M of that magnitude or greater for the particular type of effect that's being considered. This can be calculated in a manner similar to a reference dose, but using distributions instead of fixed factors, and then making explicit and transparent that we're talking about a specific magnitude effect at a specific incidence. Obviously we can't estimate this with infinite precision, so these are always going to be have a distribution essentially to reflect the uncertainty. And if you want to have a sort of a conservative estimate, then you can pick a point on this distribution. So just to illustrate this a little bit more explicitly, so the reference dose, you might have a NOAL divided by 2 uncertainty factors. In the WHO parlance, they're called assessment factors. That's why it's AF instead of UF for interspecies and intraspecies extrapolation, from a rodent study to humans, and then from a typical human to a sensitive human. So in this case, for a NOAL, for instance, that doesn't really specify what magnitude of effect we have. It will depend on the sample size, depending on the amount of variability in the system, what you would declare to be, what magnitude effect would actually be associated with that dose. And also the fraction of the population that you're trying to predict is not specified. If you apply a factor of 10, we're just sort of kind of quasi-tautologically saying we're protecting those individuals who are within a factor of 10. So, whereas transitioning to the HDMI, we are really talking about a specific magnitude effect by using a benchmark dose. We're talking about being explicit as to what these interspecies factors are, sort of a physiological one, for instance, body weight to 3/4 for interspecies for oral dosing, or using sort of regional dosimetry for inhalation, and then also any residual uncertainty due to chemical specific toxicokinetics or toxidynamics. And then also we're being explicit as to what sort of the fraction of the population we're aiming to protect. So then where do tissue chips fit in? How do we calculate sort of dose response on tissue chips? How do we make these extrapolations from the tissue chip context to an in vivo human who's exposed to some toxicant? And then how do we address the variability question? So really, in terms of addressing the uncertainty, the challenges for tissue chips are fairly straightforward in terms of looking at dose response. How do we take this to a whole human quantitatively? And then what is the degree of uncertainty associated with the data from these type of platforms relative to the cost and throughput, and then in comparison with in vivo data or in comparison with other in vitro systems? So the third challenge is really addressing human variability and susceptibility. And here, there's sort of multiple motivations here. First, from sort of the stakeholder and decision makers and affected communities, variability is an extremely important consideration. And that is often one of the drivers of initiating or how we want to make decisions in a particular context. However, there's really few examples of addressing human variability in human health risk assessments beyond applying sort of tenfold factors. In pharmacokinetics, there has been some efforts in population variability, looking at population toxicokinetic variability. If you have epidemiologic data with good stratification, you can get some idea of sort of different risk groups. But beyond that, really, it's just dividing by 10. And then even on the experimental side, toxicity is usually still evaluated in test systems, whether it's in vivo or in vitro, that are homogeneous in all their aspects, including genetics. So, this concept here is really derived from work from a previous workshop in this series, in which this paper by Zeiss et al., Lauren sitting in the second row here, was developed as a sort of a follow-on to that workshop, and it's that in addition to source to outcome, the sort of the typical source to outcome continuum, you really have to layer onto it biological variability in all these aspects. And that biological variability can take the form of, the genetic variability, which there's been a lot of discussion about, as well as, epigenetic effects, gender, life stage, pre-existing health conditions like comorbidities, co-exposures to other compounds, food and nutrition status, and even non-chemical sort of psychosocial stressors, which have been shown to affect people physiologically in a measurable way. And these different types of variability can really modify the source to outcome in two different ways. One is by modifying sort of the parameters of the system, how the system will react to a perturbation, and then second, by modifying their baseline condition. Where are they starting out? Are they already compromised in some way due to some other stressor or some other genetic factor? And it's really these two things together, source to outcome and biological variability, which together create what is the population impact of a chemical exposure. So this concept of these two types of variability, I'm going to just dwell on for just a minute. So parameter variability is kind of, I think, what most people think about in terms of variability. And that's looking at toxicokinetic parameters like Vmax or receptor binding affinities, or how responsive signaling cascade is. And that is really at modifying the shape of the sort of dose response or internal dose response or whatever whatever part of the source to outcome continuum you're looking at, sort of modifying how you get from one box to another. Baseline variability, on the other hand, is a change in the initial conditions of the source to outcome relationships, and that might be the endogenous ligands for biological responses, variability in that, co-exposures to common metabolites, physiological status in terms of functional reserve, things like that. And that kind of conceptually you can think of as it's where you're starting out on the dose response curve. Some people might already be partially up a dose response curve because of co-exposures, and so in the environmental decision-making context you're thinking about, whatever regulatory decisions you're going to make, they're going to affect, one part of they're only going to affect, an incremental dose, and different people might already be, having a background dose that you're not going to be affecting in your regulatory decision. And so in looking at the impact of different regulatory options, you would need to consider the fact that people are already starting on different parts of the curve. So this can be incorporated into sort of this HDMI concept and that's what, as part of the WHO project, we've looked at historical data on human variability. And most of this data is actually available on toxicokinetics. There's a lot of kinetic data on variability in pharmaceuticals and some on environmental chemicals. And in terms of overall variability, really only Dale Haddis at Clark University has done some effort in compiling sort of what is the aggregate variability in terms of the dose required for a particular individual to respond. And what we did in the WHO group is we compiled this data and looked at what is the distribution across chemicals across endpoints, and found that depending on what percent of the population, and there's substantial variability compound to compound in terms of how much so there's variability in the variability. For specific compound, there will be X amount of variability in the human population in terms of a response to a toxic response. But for a different compound, that variability might be less or more. So without chemical-specific data, all we can do is we can say, well, we hope that the chemical we have is drawn from the distribution of existing chemicals. And this basically shows sort of this, distribution across chemicals of what factor you would need to cover 90, 95, 99% of the population. I deleted the 99.9% just because to unclutter the graph. But so, for instance, to cover 95% of the population, that's the red line, and really, on average, maybe a factor of 3.5. Is sufficient, but some compounds are beyond a factor of 10, and some compounds are less than a factor of 2. So without chemical specific data, that's really the best you can do. But even so, we can still move forward to make decisions because we can capture that uncertainty in this sort of HDMI approach. So another area that maybe can parallel with sort of the tissue chips, and that's already actually been discussed in the first talk, is to looking at human variability using genetically diverse experimental systems. And in terms of the mouse, the collaborative cross is an example of one sort of resource that has controlled and reproducible genetic variability in a population of mice. And then 1,000 genomes is another example of sort of a library of cells that are drawn from a diverse human population. Just as an example of sort of proof of concept for trichloroethylene toxicokinetics, I worked with Dr. Roussin and some other colleagues to look at looking at different mouse strains in terms of how variable they are in terms of their metabolism of trichloroethylene. And so this first panel is the B6C3F1 mouse, which is the typical mouse that's been used in bioassays, which provides sort of a baseline for what the concentration profile of the metabolites are after a gavage dose. But if you look at other strains, you can clearly see variability, particularly in the bottom two, which are products of GSH conjugation. So, if you look across strains, look at the top panel there on the right is the oxidative to GSH conjugation ratio. And, obviously there's much more oxidation than GSH conjugation, but still, in terms of the variability across mouse strains, there's clearly, about an order of magnitude difference. And also on the bottom there is the amount of trichloroacetic acid that's produced, and here it's much tighter. There's much less variability. Now, how does this translate to potential human variability? Well, so we applied a population PBPK model to these data and examined, what's the predicted amount of interstrain variability in the mouse for a various number of sort of internal dose metrics, and found that really they actually overlapped very nicely with the amount of human inter-individual variability that was derived from actual human toxicokinetic data. Since trichloroethylene is one of the few environmental compounds which there are actual individual human data on of the toxicokinetics that's been studied over last 50 years or so. So in the mouse population, variability across these strains was actually nearly identical to what was predicted based on human data. And so where potentially tissue tubes might be able to serve as sort of instead of a mouse, we would have populations of tissue chips essentially, and maybe be able to also perform that extrapolation into how much variability there might be in a human population. In terms of toxicodynamics, this is an example out of Dr. Roussin's lab for looking at in vitro cytotoxicity across many cell lines. So there's across over 1,000 human cell lines from the 1,000 Genomes Project. As you can see from these three compounds, the amount of variability in this sort of in cytotoxicity is quite different across compounds. On the left, the compound on the left has very little variability in what the cytotoxic dose is, whereas the compound on the right has extremely large range of variability, over 100-fold from the 5th to the 95th percentile. And then this was repeated with 179 compounds, and showing that as was shown in the more limited sort of in vivo human data, there's substantial chemical to chemical variability in the amount of human variability. So the blue histograms are the actual empirical data, and then taking out sort of the technical variability to the measurement system, that red line is derived. So maybe potentially tissue chips could also be a screening tool for this type of analysis. So in terms of addressing variability and susceptibility, what are some of the questions and issues with that tissue chips might be able to address? First and foremost, and that this has already been mentioned today, is that having some direct estimates of human variability using experimental systems with controlled variability, looking at a population of human cells to populate these tissue chips. A bigger challenge may be to look at sort of non-genetic variability, but here, maybe induced disease states and varying the, nutritional or the nutritional factors in the serum or that's used, might be ways to look at that. Another potential question is whether, combining with computational models in the way that, combining the interstrain mouse variability with a population PBK model, could that be a more powerful approach combined together? And then also the issue, of course, of cost and throughput in comparison with getting in vivo data. And then finally, whether we might be able to better assess some of the more downstream variability in terms of actually looking at actual adverse outcomes, and not just sort of by early upstream biological responses. The last challenge is really supporting economic benefit cost analysis. And so first I wanted to provide a brief description of what economic cost benefit analysis is in a regulatory context. It's really a way to, because there's a lot of sort of general misconceptions as to what the economists are actually doing in doing these regulatory analyses. The aim is really to stimulate a private market in reduced health risks and costs of regulatory actions, and to attempt to capture these in dollar terms. And in many cases, these are actually required either by statute or by executive order, and so there's no way to really avoid that. Now, unlike sort of the more popular misconceptions, this isn't sort of putting a value on an individual's life. This is putting value on small increments of reduction in risk. So it's like, to take an analogy with sunscreen, like people buy sunscreen, they're willing to pay for sunscreen in order to reduce their risk of getting melanoma. How much are people willing to pay for sunscreen? Well, we know that because sunscreen's on the market, people, are pay, and there's different, SPFs and different types of sunscreen, and so there's a private market that we can value what people value that reduction in risk is. But for environmental chemicals, there really there's no private market in it. And so it's really trying to value, if we reduce PM by a small amount, by amount, and we're reducing a risk by a small amount in the population, what is that? What are people willing to pay for that? And usually this is supported by epidemiologic data, because that's, for instance, PM and incidence of heart attacks, say. And that epidemiologic data, you take, fit a regression line to it, you put that into sort of what's called a health impact function, which is a measure of what is the expected change in incidence in your exposed population due to an expected, a regulatory change in the amount of PM. And then you value the economic benefits of that change in incidence, either by willingness to pay or by what's called cost of illness. Now, one of the issues is that things like a reference dose or reference concentration can't be used to support economic benefits analysis. All it is a single number. So you can't really derive what is the impact of different levels of concentration of a compound from a reference dose. On the other hand, the HTMI approach can actually do that, because we're actually deriving, the incidence of that's what the I is for, the incidence of a particular magnitude effect, which then can be used to derive a health impact function. So then the question is, goes back actually to dose response. Can we use tissue chips to develop these sort of dose response relationships, or even better, a population dose response relationship that can be translated into a sort of a concentration response in terms of health impact that can be used by economists to value those changes in exposure concentrations. So then how can, so, this is basically the questions I just asked. Can you use tissue chips to establish an in vivo human dose response function? Can you quantify sort of is the tissue chip response something that you can quantify economically, or do you need some sort of further downstream model in order to take the response in the chip to something that would be considered an adverse outcome from an economic valuation point of view? And then ultimately, of course, what is their cost and throughput relative to available in vivo data? So I know that was a lot in a very short amount of time. Just to sort of quickly summarize, sort of the basic questions: where do tissue chips fit in the source to outcome continuum? What levels from media concentrations all the way to physiological responses are they interrogating? How might they be incorporated into our standard sort of hazard identification and dose response assessment approaches, whether in identifying health effects, whether in deriving points of departure, et cetera? And then finally, what are some of the environmental health challenges that tissue chips can help address? Because ultimately, we're interested not only in source to outcome, but also population variability to be able to assess what is the overall population impact of particular chemical exposures and the benefits of reducing those exposures. Thank you very much.
So let's take one question, please.
Yes, I'm interested in the different factors leading to the definition of an exposure. There are environmental concentrations, there are occupational concentrations, there are also acute versus chronic concentrations. I have been concerned with massive spills of very hazardous chemicals in populated area from maritime transportation.
Mm-hmm.
More recently, I know of work being done by Department of Homeland Security and specifically by EPA on potential threats from natural or antagonist sources to the water supply, for example. So in cases where you have a massive sudden exposure to a geographically confined population, do you, and various scenarios about how those might develop, do you see tissue chips as being a useful mechanism for evaluating those hazards as well?
Potentially, if they can be, sort of ruggedized and you take the actual chip out into the field, kind of like, doing, real-time on-screen sampling. I'm not familiar enough with the chip technology to see whether that, is a potential feasible application. But, certainly, having a more full physiological system to take out and really, so, there's some mixture of chemicals that have suddenly been released. If you could take that whole mixture and expose a tissue chip to it, that certainly could be a potential application. Thank you.